Okay, so today we're going to talk about the sliding filament theory. The sliding filament theory has a myofibril, and in the myofibril it has an actin, which is a thin filament, and it's represented by these blue lines on this little structure, and these contain myosin with binding sites included. It also includes two regulatory proteins. These proteins are called tropomyosin and troponin. These also contain myosin heads that have actin binding sites with individual molecules. These molecules also have heads called cross bridges between actin and myosin that also contain ATP binding sites and ATPase enzymes. And these enzymes are also used within the sliding filament theory to represent or demonstrate the relaxation and the contraction of muscles. Okay, so now we're going to actually talk about how the sliding filament theory occurs. It occurs within the myofibril structure, which is also called a sarcomere. Here we have a thin filament and a thick filament, which are different within structure and size. This is the myosin, and this is the myosin head. Attached is an ADP phosphate. The myosin head with the cross bridge attaches to the actin filament, and what happens is that the ADP phosphate are released within the sarcomere, and this causes a working stroke. The myosin heads that are used bend sliding towards the enzymes in that angle. The ATP then comes in, and the myosin then becomes low in energy during this working stroke. With new ATP, this causes detachment from the head. Because ATP is hydrolyzed and split, it turns out into ADP and a phosphate. ATP is split, causing the myosin to move towards the thick filament, and this is what causes the cycle to occur all over again. Once again, demonstrating the contraction and the relaxation of a muscle, and that is the sliding filament theory within the body.